Hello everyone and welcome to the tutorial. In this session we will see how you can use BLAST for identifying any organism that you have isolated. So let's get started. So first you have to open your browser and type NCBI BLAST. Open the first link. On the home page you will find that you can either BLAST two nucleotide sequences, two protein sequences, a translated nucleotide sequence against a protein sequence and vice versa. Then again, if we go down, you can see that Blast can be used as a standalone tool, as an API, and also from the cloud if you are low in RAM. Lastly, there are certain specialized searches that you can use. For example, comparing two sequences across the entire span or finding conserved sequence. So coming back, in this video we will be covering how you can blast two nucleotide sequences against each other. So for that, click on this icon. Here in this box, you have to enter either an accession number or a gen info identifier or a FASTA sequence file. So I have a FASTA file that I will be copying here. So the file is in txt format. Open the file and copy the sequence. Then again go to the BLAST website and in this box just paste it. Automatically, the name after the FASTA header symbol will be used as a job title. Additionally, you can also browse any file from your PC. Then going down, you can select from any of these databases depending on the type of sequence or the organism from which you got that sequence. In my case, I have a bacterial sequence, so I will be selecting the rRNA/ITS databases. So here you can see that it can be either 16S ribosomal for bacteria and archaea, and then 18S, 28S, and ITS for fungi. Then you can either select the organism or you can skip it. We generally keep the selection program to megablast, that is highly similar sequences, and just check this box so that the results will be opened in another file and click on blast. Now the program has run and these are the sequences or organisms that got the best match with our query sequence. So from here I can be sure that it is belonging to the Staphylococcus genera. However, the species may vary. As you can see that the maximum score, query coverage, e-value and identity are identical for all of them. So let us take a look at these values and scores. For maximum score and total score, they are calculated after scoring all the matches, mismatches and gaps that may have occurred at the time of alignment. The query coverage needs to be high. It should be around 95 to 100% in case of 16S rRNA sequencing. It basically shows how much of your query sequence got aligned at the time of alignment. Then we have the e-value. This value basically measures the probability that a given sequence match has occurred purely by chance. So the lower the e-value, the better the or more statistically significant the results are. Then we have the percentage identity. The higher the percentage identity, the better the results are, the better the confidence is. Then we have the accession length of this sequence that is shown here. And we have the accession number again of this sequence that is shown here. So this was the basic of BLAST that you need to get started with the tool. Just one thing that I would like to mention here is that the query length of my sequence is really low. It's only 131 base pairs long, and as you can see here, almost every sequence that is mentioned has an accession length of around 1550 to 1600. 16S ribosomal RNA sequence is almost 1600 base pairs long, so it becomes crucial that the length of our query sequence is higher. It should be around 1500 base pairs to reliably say that the alignments are correct. And now that you know how to use BLAST, just go and identify the organism that you just isolated.